the last class of fungi that I want you to know in lecture are the glomeromycetes, which are the arbuscular or vesicular arbuscular mycorrhizal fungi or the endomycorrhizal fungi. And so these are obligate mutualists, which means we don't see them growing without their plant partner for any length of time. They don't make it, they're obligate. And so if you were to buy some of these fungi, they usually come mixed with plant matter because they don't survive very long without their plant partner. And so when I was doing my graduate work, I worked with ectomycorrhizal fungi because at least back then they knew how to grow them in the lab separate from the plant. But these, we still don't have a way to grow them separately. And so Glomus species, that genus, the Glomus, they're the largest genus that we know of isolated endomycorrhizal fungi that have been studied. And so the Glomeromycota, that phylum, which the glomeromycetes fit under, they were credited initially with the idea that they were the type of fungus that was present millions of years ago that helped the plants go from an aquatic ocean environment to terrestrial land. And so that was the thought that they were there right from the beginning. And so this has been debated now. So with the glomeromycetes, there's certainly the VAM type or mycorrhizal fungi, but how they were involved in the evolution of plants is still, there's been some interesting additions to this idea. But I can't remember if I showed you this with they used to be called vesicular arbuscular mycorrhizae. Now they kind of dropped off the vesicular term. But the vesicular term comes from the idea that they form these vesicles, which is just fungal hyphae that is ballooned up. It looks like a balloon or vesicle. And then some of the fungal hyphae, they form arbuscles, which if you look up that word, it means dwarf tree. And especially this lower right image, look, I mean, that really does look like a dwarf tree, but that's all fungal hyphae that is in the root tissue of the plant. And so that top left image that you see there is a root cut in a longitudinal section, so along the length of the root, and then you're seeing the arbuscles in the root tissue. So the thing that is hard to see with these images that I want you to be aware of is that if you look here in this section with this drawing, the fungus is this area here, and where's the word? So periarbuscular membrane. Periarbuscular membrane. I guess I wrote over it a little bit. So even though it looks like the fungal hyphae is like in the root cell, it's not directly in contact with the cytoplasm. There's always a plant-derived membrane that surrounds it. So even if it's fungal hyphae grows, it looks like it's in the cell, it's not really in contact with the cytoplasm. All right, and that's an important distinction I want you to pick up on, because if you read the common literature, they'll say mycorrhizal, the VAM fungi hang out in the root cells. And it's like, well, no, not really, because if that implies they're swimming around in cytoplasm, and they're not doing that. They're always have a boundary of the plant cell membrane. Because if the fungal hyphae directly penetrated in and was actually in physical contact with the cytoplasm, it tends to make the plant defend itself. Like, ooh, you breached my boundary, I don't like that. And so then they'll usually kick into plant defense mechanism. And so if they do they call it a controlled infection. The plant is allowing the fungus to come into the root tissue, but it's creating a boundary and saying that's far enough. You're okay to come in, but you can't breach the boundary of the membrane. So just be aware that there's a lot of literature out there that it's a little confusing the way they have it phrased. So if you think about all the plants that are in this relationship with mycorrhizal fungi, there's a bunch of different mycorrhizal fungi. But the VAM fungi, and in particular, the fungi that are in the Glomus species, they're really, really abundant. So many, many, many different plant species have this kind of relationship with this type of mycorrhizal fungus. So I'll come back to this idea in a little bit here. But there's also, when you think about fungi, you probably hear a lot about the pathogens. That's kind of they get a lot of press. And so if someone were to ask me, would you rather get infected by a fungus or a bacteria? Especially, if a real serious pathogen, not something like athlete's foot, but just like a serious pathogen, I'd rather take a bacteria than a fungus, because fungi are too much like us. They're too similar to us. It's hard to find antibiotics that kill the fungus and don't kill you or hurt you. And so I think most medical microbiologists would agree that fungal pathogens are more concerning than a lot of the bacterial pathogens, because at least so far we have antibiotics that can take care of the bacteria a little bit easier than we can with what we have going on with the fungal pathogens. So this one, pneumocystis, it used to be called Pneumocystis carinii. They renamed it, which you don't need to know. I'm not asking you to memorize the pathogens per se, but it is, I just want to let you know that there are definitely some not so nice fungi out there that are problems for humans and other animals. And this one tends to be causing pneumonia, but it's usually people who are immunocompromised. Maybe they are really sick with something else, or they're battling cancer, or they have AIDS, something like that. Usually the late stages of AIDS disease, you usually see this kind of opportunistic fungal pneumonia type pathogen. Then there's the so-called microsporidia, which are technically, I guess, not straight ahead fungi. They're kind of weird, and I'm not going to get into them, but they're weird, and they are also with immunocompromised, and again, showing up in a lot of late stage AIDS patients that are struggling to survive. And then this one too has probably the newest, well, one of the newer ones, Rhodotorula mucolaginosa. This one is an opportunistic pathogen. It is the unicellular yeast type of fungus, and it is found it can be found normally and not causing disease, and just kind of hanging out with the human. But then under the right conditions, it can become opportunistic and create a pathogenic disease process in the human host. So they're worried about this one. Dr. Sharp and I worked with some undergrads on that. We were curious to see if some of the Streptomyces bacteria that I had frozen stocks of, if they might be capable of inhibiting Rhodotorula. And so we were doing some cold plate studies where we were looking at spot inoculating Streptomyces after we did a fresh plate of the Rhodotorula yeast. And then we would spot inoculate the Streptomyces and incubate and look for the kill zones or inhibition zones. That's the Streptomyces that naturally forms that blue-like pigment. And you can see some nice inhibition zones against Rhodotorula. So the reason we started looking into this is a lot of the literature was saying we can't find an antibiotic that can really make a dent. And so we were interested in, we knew that a lot of the Streptomyces I had were capable of antibiotic production. So got some really nice study or results with that study showing some really large kill zones. So there is the capability to find some kind of antibiotic, but it's going to take some time to get that, to the point where people can take an antibiotic for this type of infection. So if you go to the CDC website or something like that, you can see they usually have a list of emerging fungal pathogens or fungus-like pathogens, and you can see that there are a range of different types of fungi. And, but usually when you look at, especially with humans, it's usually this idea that you need to be immunocompromised for the pathogen to be successfully causing disease. But you can see, it's not just humans worried about, of course, honeybees with colony collapse disorder. There's a the Nosema type Microsporidia have been implicated in that, as well as, plants. The number one pathogen group against plants is fungi. There's more fungi than bacteria that cause problems in plants. But on the other hand, there's all the mycorrhizal fungi that are super beneficial for plants. So there's a lot of good fungi out there too when it comes to the plant world. Ever seen this documentary, Fantastic Fungi? So that one, it was on Netflix for a while, but I think they pulled it now. They rotated it off, unfortunately. But there's a book. I have the book, very beautiful full color, photographs, beautiful imagery. The documentary, I think, is worth watching. And now if you go on to like YouTube, you can see all kinds of really cool videography where they speed up the growth of the fruiting body. And it's just really some really talented videographers out there now that are starting to use fungi as their model to do some cool artistic videos. So check that out when you have a spare moment. There's a lot of really nice videos that have been posted. Okay, so I wanted to shift into the second slide set on fungi. Still have a few things I want to say. I wanted to just talk a little bit about kind of like the ecology of fungi and a little bit of how we're, using them. What happened there? Didn't mean to do that. So this is part 2. I like this quote. All mushrooms are edible once. And the other one is a good one too. So I looked high and low for a poem about fungi. There's not that many out there. So if you have an artistic writing ability, you might want to publish your own mushroom poetry. But if you are bored and want a little poetry about mushrooms, there's one for you. I think it's a pretty good one. I won't read it out to you. You can do that on your own. Do you remember the Got Milk ads? The ad campaign Got Milk? So that's a play on that. Got Mold. So that's Kevin working in the greenhouse. He did many years ago, he did an undergrad research project on edible fungi. He designed and built his own fungal incubator. And a lot of the times when you work with growing the fruiting body, you have to have really nice humidity levels and a way to control that. So he created this humidity tent. He even inserted a zipper in it and everything. It's like, he knew how to sew better than I did. And he had that set up in the greenhouse, and he had really interesting results. He used different media and substrates in order to grow these fungal cultures all the way to the fruiting body. So anyways, so Got Mold. So I wanted to talk a little bit about how we use microbes or how the ecosystems benefit from the presence of fungi. So one of the big ideas I want you to get from this, I'm not going to go into details, but this idea of carbon sequestration, you've probably heard a little bit about that, with the increasing levels of CO2 because of climate change, they're kind of scrambling to figure out how can we try to mitigate that. And so one thought is to look at the mycorrhizal fungi's role in this process, and the results are very promising that they can be significant in terms of sequestering carbon into the soil system, which basically you're locking the CO2 into biomass underground, which is going to mitigate CO2 levels in the atmosphere. And so this is one area of research that they're really looking at more carefully and saying, how can we promote this or augment this to try to deal with the increasing CO2 in the atmosphere. So that's one idea that's a hot topic right now. And just in general, fungi are awesome at the whole idea of carbon cycling on Earth, in our planet. And I'm not going to get into the details of that other than know that they're a major player in the idea of carbon cycling. And if you've not had intros to ecology in a while, this is just trying to show the cycling of carbon with really fungi are huge players in this, but so are bacteria. Very, very important that the microorganisms are helping out with that, but definitely the mycorrhizal fungi are big in the carbon cycling realm. And then the idea that you have the presence of mycorrhizal fungi millions of years ago, helping the plant make the transition from the ocean to land. That's a pretty well-established and accepted hypothesis. And so this is a paper published about that. And so, we're talking about the Ordovician era in the time frame of evolution. And so they're debating about which type of mycorrhizal fungi. There's been some new evidence that there's this other group called the Euchroomycotina. But those are mycorrhizal too. They're the endomycorrhizal type. And so I highlighted just some of the idea that in order to get this transition from the ocean to terrestrial and for plants to make that transition, they think mycorrhizal fungi, and particularly the endomycorrhizal fungi, had to be present. And so this is, if you think about it, a huge transition, a very difficult transition, going from totally aquatic to very different. The terrestrial experience is certainly very, very different compared to the ocean. And so there's still, I wanted to put this in here just to show you there's still debate about what type of mycorrhizal fungus. However, it doesn't seem like they're disputing the fact that there were mycorrhizal fungi there. And so that's been established. Now they're just fine tuning who exactly might have been there and the major player. But if you haven't taken a botany class, basically they think that green algae or this other group called the charophytes, which used to be part of the green algae and now they separate them. So they're really closely related. So the green algae or the charophytes were the ancestor to the terrestrial plants, is the thought. And getting that shift over onto land is a huge, amazing shift where so much had to go just right. And they have also documented that there are, in the green algae, there are genes on the chromosome that allow for this mycorrhizal relationship to kick in. And so this idea that if some of these plants made it onto land and started expressing those genes where the fungi were there, you could get a partnership and allow for the plant to be able to, gain a footing, so to speak, on terrestrial habitats. And so this is about 500 million years ago that they think this was going on. And so it's pretty well established that the accepted hypothesis that the mycorrhizal fungi were there. And they're still debating which type, but some organism, some fungus from mycorrhizal fungus from the Mucoromycotina or the Glomeromycotina phylum. So still debating it, but they know, they're pretty confident it was a mycorrhizal fungus through the fossil record and also studying the DNA of the green algae and the charophytes having the genes that allow for this mycorrhizal symbiosis. So if you look at it, so far, this is a paper that was published in 2018. You can see the pie chart and the endomycorrhizal type fungus wins. All that blue is the VAM fungi or the arbuscular mycorrhiza, endomycorrhiza. And so the vast majority of plants have that type of association. And then you see the ectomycorrhizal much smaller. And then you have some of these other groups I haven't really talked about, the orchid and the ericoid. And then there's just a small bit that are non-mycorrhizal in the sense that there's some plants that just seem to never form this relationship and they don't know exactly why. And then this orange piece of the pie is arbuscular mycorrhizal fungi shifting to non-mycorrhizal. So it sounds like it's kind of like this ephemeral relationship that doesn't last long and then it fades and then it comes back and fades on some of these plant species. So, but certainly you can see that the arbuscular mycorrhizal type is very, very successful and been able to form a lot of partnerships. So just going back a little bit to this idea of the different groups. So with the ectomycorrhizal fungi, that smaller group, they're still really important to a lot of different plant species. And you can see that their benefits to the plant is that the mycorrhizal fungi really help them get access to, in particular, nitrogen, phosphorus, and potassium. I'm surprised they didn't put potassium in there. And then it's the same with the arbuscular type too. They're showing lots of nice acquisition of minerals that the plants need. And you can see that with the ectomycorrhizal relationship, it's a little bit more long-term in the sense that one species will hang on to this relationship with the plant for a couple of years. If they study it in the lab, some of the fungal hyphae is more ephemeral with our arbuscular type. But like I was saying before, with the arbuscular type, the fungus does not cross the cell membranes. Okay, so the plant membrane is always surrounding the arbuscule fungi or the vesicle parts that look like they're in the cell, but they're not truly in the cytoplasm. Does that make sense? Okay, so, and then with ectomycorrhizal, so a lot of the plants that you see, a lot of the trees that you see around here, you're gonna see that they would have the ectomycorrhizal type of interaction. Whereas the arbuscular, some of the tropical trees have that, and maybe some of the temperate trees and shrubs, but arbuscules are mycorrhizal fungi. They tend to form a lot of relationships with like the crop plants, non-woody plants, the grasses in your lawn. So, there's a lot of interactions with those types of plants. And some of the major differences between the ecto and the arbuscular is being shown here when they interact with the roots. So the ectomycorrhizal, reason they're called ecto is that they form this really thick, dense growth over the surface of the root that you can see with the naked eye. If you train your eye to it, I'll show you some pictures. And then they can grow their hyphae in between the root cells, but they're not penetrating the cell wall. They're just in between. And so that is typical. And then of course they have the fungal hyphae that grows off the root into the soil. And then with the arbuscules, they don't have that thick mantle, but they grow on the surface, but just not real thick like that. And then they can grow in the root tissue, but they're a little bit more intimate because the arbuscular type can make it past the cell wall of the roots, but not penetrate the cell membrane. So they're kind of in between the cell wall and the cell membrane, and they can kind of like hug up to the cell membrane and push against it, so to speak. So it looks like they're inside, but they're always covered by that plant membrane. So the arbuscular type have a more intimate interaction with the plant root. But the ectomycorrhizae, you can see with the naked eye, if you look at some roots, like if you're out hiking and you see a overturned tree, go look at the root system. I bet you'll be able to see this. If you look at it, these are magnified slightly, and this is like under a dissecting scope, except for that. And so you can see that the root here, like that orangey part is the root. And then the more pumped up whiter portions, that's the root covered by the ectomycorrhizal fungus. So it looks plumper, thicker. And then that very often, especially around here, you'll see many of the roots will shift into a Y-shaped growth. Usually roots are just straight, but they often do a Y shape. So you can see a little bit here too, that Y shape to the roots. That's classic. If you see that, you know that that's a ectomycorrhizal root. And then here too, you can't see it really well from where you're sitting probably, but some nice Y-shaped lateral roots here. This is more like what you would see with the naked eye. So they're small, but if you look carefully, you can definitely pinpoint that the roots are associating with the ectomycorrhizal fungi. And I like this picture too, because it's showing that you have all the hyphae coming off. You can imagine all that hyphae off into the soil, and it's scarfing up all the lovely minerals and water that it can give to the plant. So look for that if you ever see an overturned tree, or if you dig up some roots. Endomycorrhizal, you're not going to see unless you do some special staining. Just your action of, taking the roots out of the soil system, you're going to rip away all the surface hyphae because it's so thin. So you need to do some staining. So I've had students like in plant phys when they are doing their research projects and they are testing mycorrhizal fungi. They'll do some staining at the end of the experiment where you can use trypan glue, and it'll stain the fungus, the fungal hyphae, but keep the root unstained, so you can clearly see that the root has been colonized by the mycorrhizal fungi. And so that picture is showing the arbuscles and the vesicles along the root. And then this is the top right image. That's an electron microscope picture showing the dwarf tree, the arbuscle type growth pattern, but that's all just fungal hyphae. So it's definitely a win-win because the plant gets lovely nutrients, extra access to phosphorus and nitrogen and potassium, lots more water availability because the fungal hyphae is nice and thin, much thinner than the roots, so they can get into cracks and crevices that the plant roots just can't get to. So all of that increased access to minerals and water, it's going to stimulate the seedlings. They're going to grow really well. They're going to thrive. They're going to get protected as well because the ectomycorrhizal, because they form such a thick growth along the surface of the root, they can physically protect, but they can also chemically protect. Both types of fungi can produce antibiotics and other chemicals that can help protect the plant against pathogens. And for the fungus, they get fed. The plant feeds them. So the plant will do photosynthesis, make all this lovely sugar, and then send that sugar to the fungus to support its growth. And some evidence too of growth factors that the plant can make that can help the plant. So some studies have shown with certain ecosystems, forested ecosystems, that some of the larger trees can send 30 to 40% of their sugars to the fungus. That's a lot. And so obviously the plant thinks, so to speak, thinks it's worth it to share all that sugar with the mycorrhizal fungus. The payoff is worth it. So you have this relationship is really gaining interest in the agricultural industry, whether it be for traditional crops or medicinal crops. The idea of making sure that there's a nice, healthy network of mycorrhizal fungi in the soil system, and even hydroponics. There's evidence that mycorrhizal fungi are happy in a hydroponic setting. One of my grad students was looking at that with she was growing spinach crops, and the mycorrhizal fungi were being mixed into that. And so this idea that you need to pay attention to the mycorrhizal fungi to grow, crops that are really going to be able to make it all the way to harvest and get what you want from them, whether you eat them or medicinal properties. One of my former grad students worked with orchid mycorrhizal fungi and orchid endophytes. And so this idea that you have orchid mycorrhizae, this is really funky because the group of plants that the orchids themselves, they're super abundant across the face of the earth, even though we don't have many up here. It's kind of weird. We don't have that many, but if you go into the tropics, they're everywhere. And so they're hugely successful, which I don't understand really, because you look at their seeds and they literally are like a speck of dust. Most seeds have a nutrition source in the seed to help the baby plant grow. Orchid seeds don't do that. They have to have, in fact, a particular mycorrhizal fungus. Right where that seed lands, there has to be the right fungus in order to get that seed to germinate, because the fungus does the feeding, essentially. The fungus is like the mom that, is feeding the orchid baby as that seed germinates and grows. If that fungus isn't there, that seed will never germinate. And so they have to rely on the fungus to help with that. And so that's why years ago, they didn't know how to grow orchids. And so they go and hunt them. They were orchid hunters. The rich people back in the Victorian period in the 1800s would send people out around the world, go hunt me some orchids. I want that in my, my collection. And they bring back the orchids and have this, they'd be able to boast that they have this fungus in their collection. And it was a thing. I mean, people would go all over the world. There's documentation that orchid hunters would kill each other over certain orchids because they were so rare and coveted, and they were getting paid so much by their rich person back home. Yeah, there's some interesting stories. There's a book written about orchid hunters that if you get bored in the summer, you should read it. It's an interesting phenomenon. So you gotta have the right seed, the right mycorrhizal fungus for that orchid species. And so they finally figured it out. It took a while, but they finally figured that out. And now they know how to, work with orchid seeds, but it's a very specific interaction. So, it's still a thing. And if you know anyone who's into orchids, it's kind of a fanatical love that they have for orchids. And there's I'm sure, $1 million industry because they are such an interesting plant with amazing flowers and design of flowers. So when you look at the fungus, though, once the orchid has established and you look into its root tissue, they have lots of fungal hyphae in there. And so they are really, the orchid roots are really well colonized, by the correct species of mycorrhizal fungus. So interesting process. They're also looking at mycorrhizal fungi and their ability to create a stable ecosystem. This is one of the more catchy titles to a published paper, with the idea that they can facilitate communication, learning, and memory in trees. And so this is Dr. Simard's work in British Columbia. She's probably one of the big pioneers here in America with this idea of how important the mycorrhizal fungi are for forest ecosystems. So we talk a lot about her work in my advanced botany class that's being offered this fall, by the way. And so this idea that you have all these mycorrhizal fungi underground and their hyphae connect all the different plants in the forest ecosystem, and that allows all kinds of nutrient flow amongst the different trees, even amongst different species. And so they can document that you can have so-called hub trees. So the darker and bigger the color means that you have more mycorrhizal connections to that tree. And so there are so-called hub trees. She sometimes refers to it as mother trees. Avatar, the movie Avatar, kind of, there's an interview where she says they must have read my research, because that's kind of what she was getting at with the mother tree and the connections underground. So this is from one of her papers. And so all these connections amongst the different trees, and some are more connected than others. And the idea that you can have flow of nutrients or certain chemicals from these hub trees in really high amounts to some of the other trees, even if they're not related. So they can be unrelated. And they've been able to document, quite a bit of a flow of different chemicals, including water, defense molecules. They are looking at kin recognition information amongst the different
species in the forest. So you don't have to write all this down. I'm just trying to show that there's a lot of research going into this, looking at what are these mycorrhizal fungi doing for these forested ecosystems, and how are they controlling the flow of information in any way? Or are they just a passive thing? And it's still debatable, but there's nice studies out there showing that the mycorrhizal fungi can somewhat choose where some of these nutrients go to. They're exploring that a little bit more, but they're realizing that what's going on underground may be having a pretty major impact to how that forest develops over time. And so that's something that's fairly new, and we know a lot about above ground things that are going on with, plant succession. But the mycorrhizal fungi have been overlooked until fairly recently. Now there's some really nice studies coming out saying, hey, they're major players. They're affecting what's going on that we see, visually above ground. So let's dive a little bit more into the plant. Fungal endophytes are a thing. So there's also bacteria that are endophytes, but since this is a fungal section, we'll just focus on the fungi. So mycorrhizal fungi are not I don't consider them endophytes, because if you look up the definition of an endophyte, that means within the plant tissue, pretty much solely within. And mycorrhizal fungi are in the plant tissue, but they also grow out extensively into the soil. Endophytes don't do that. They're always in the body of the plant, and they live their whole life pretty much that way, and they help the plant. And we're finding more and more that these endophytes, very similar to all the microbes that are in you right now, that coexist with you long term, that they're doing very helpful things for you. And so if you are an endophyte, you have a pretty sweet life. You're sheltered from all the competitors around you. You're held into the plant body in the tissue. You're getting access to nutrients from the plant. What is the plant getting? Well, the list is increasing. I'll just give a few examples, but basically there's all kinds of ideas and published studies showing that the presence of the endophytes help defend the plant against pathogens, against herbivores chewing on the plant, abiotic stress, too much heat, too much drought, salt stress, things like that, heavy metals, and helping the plant with its nutritional needs as well. And lots more going on there. This is a hot topic right now, endophytes. They're looking, because it's kind of an offshoot of the Human Microbiome Project, where we're trying to figure out what are all these microbes doing for humans. And then the plant biologist said, well, wait a minute, this is probably going on in the plants too. And so they looked and they're like, yeah, we found lots of endophytes. What are they doing? And so this is a really hot topic now, looking at how they're impacting plant growth and their ability to thrive, and all the secondary metabolites that plants are making. They're now realizing, hey, those secondary metabolites, they're actually being made by the endophytes. So it's a little upsetting to plant physiologists to find out that there's this whole other world of microbes that are contributing to all these cool secondary metabolites that was attributed to the plant. Now they're realizing a lot of that is actually due to the endophytes. So here they're looking at biocontrol agents of these endophytes, and this paper was on cannabis, and looking at their ability to help the plant defend itself. So my grad student Duncan, he's doing work looking at this idea of using biocontrol agents, and we're asking the question of, if we apply biocontrol agents during the early stage of the growing season, can any of those go endophytic and help the plant long-term against fungal, and primarily fungal pathogens? But this idea is gaining interest. More and more scientists are looking at this idea. How can we use these endophytes to help the plant thrive and produce what we want them to produce, whether it be some good quality food or different medicinal properties, secondary metabolites. So this is really started back with this drug. So this is kind of what spearheaded the idea of we should hunt for endophytes and see what kind of secondary metabolites they're making. This drug is called, the common term is Taxol. And if you know anyone who has breast cancer, that may sound vaguely familiar. This is a billion-dollar anti-cancer drug. And you can see from the chemistry of it, they didn't know how to make this, but they isolated it from yew trees. And it was in the bark area of yew trees. They discovered this chemical. And it can put certain types of breast cancer into total remission. So it's very, very effective. And so that's why it's a billion-dollar drug. And so plant biologists were getting a little worried though, because yew trees were getting cut down left and right. And they're like, we're gonna have no yew trees at this rate. And so endophyte researcher, Dr. Strobel, who was at the time, I think at Montana State University out west, he said, well, maybe it's not just the yew tree that's making that Taxol drug. Maybe there's a fungal endophyte. So he and a colleague went and sampled a bunch of yew trees and discovered that there's an endophytic fungus in the tree trunk, in the stem, in the bark area, that is producing this drug. And so they're like, oh, this is awesome, because now we can just isolate the fungus. Much easier to grow a fungus than wait for a yew tree to grow, and get enough Taxol from it. So you can isolate this fungus, grow it in huge vats in the lab, and get it to pump out this drug all day long once you figure out how to make the fungus happy. And that's what they do. So now they're making it that way. And they're not sure exactly, like they know the yew tree can make it, and now the fungus can make it. So they think that maybe there is genetic horizontal gene transfer going on one way or the other. And right now they're kind of leaning toward, they think it went to the fungus, because when they work, the particular fungus is Pestalozziopsis. And when they work with that fungus in the lab, it is so easy to introduce genes into. It's like it's really happy, like no problem. It can take all these genes and it's very easy to transform. And so they wonder if the tree kind of gave, through horizontal gene transfer to the fungus, and then kind of delegated, have the fungus do it, and the tree doesn't have to do it so much. So this idea of fungal endophytes, Dr. Strobel went on a big hunt all over the world looking at these endophytes, because sometimes these fungi make the same thing as the plant they're residing in. Sometimes they make unique drugs that the plant doesn't make. And so they're really interested in them for drug discovery, as well as, of course, what are they doing for the plant? So lots of beautiful different fungi he's isolated. He's traveled the whole world looking for different endophytes. Must be a nice job to have. Just travel the world and go into really interesting, he focuses a lot in the tropical areas, looking at rainforests. But anyways, all right, we'll stop there and get into the last bit of fungi and then get into viruses next.